Hi guys, in today's topic we talk about the three protein couple receptors in our cell, in our tissue, in terms of pharmacodynamics. So what pharmacodynamics means is what the drug do to our body. So first, there are three types of three protein couple receptors found in our cell membrane. The first one is the GQ. We have the GQ, GS, and the GI. So first, I'm talking about GQ first. So when forgot the top. I'm gonna tell you the GQ protein receptors are found in the alpha-1 receptors, which cause muscle constriction and mydriasis, the direction of the pupil. Okay, so when the alpha-1 agonist binds to the alpha-1 receptors, what it do is it release the GTP, so GDP. So normally GDP, so normally GDP is bound to the GQ, but when the alpha agonist binds to the receptors, it releases the GTP from the G protein, GS, GQ, G protein, and then the GTP binds the GTP. Different GTP and GDP. So the GTP in the cell membrane will bind to the GTP, the G protein, and what the GTP do to the G protein is it truly activates the the G protein, and the activated GQ protein goes and activate the PLC, phospholipase C, found in the cell membrane. So what the phospholipase do is it breaks down the PIP molecule found in the cell membrane into DG and IP3. So DG is a molecule that activates the protein kinase, especially the protein kinase C, specifically the protein kinase C, and the protein kinase C will phosphorylate the proteins and initiates a cellular response. So DG acts as a secondary messenger. Protein kinase acts as a secondary messenger that phosphorylates the proteins, such as enzyme or the enzyme or the receptors in the cell membrane, which can cause the influx of ions like calcium ions, chloride ions, and then the cellular responses will be initiated. So this is what the DG will do. So another molecule from the PIP. PIP2 is the IP3. What the IP3 do is it evades the intracellular calcium ions, release the calcium from the interplasmic reticulum of the muscle fiber. So I'll show you what the calcium do to the muscle fiber, to the muscle. So this is the actin filament found in the muscle fibers. So when the calcium, so the actin consists of several molecules such as the tryptomyosin. The yellow color one is the actin, and then the tryptomyosin, the blue one is the tryptomyosin, the pink one is the troponin. So the calcium will bind to the troponin, and then the troponin is actually binds to the tryptomyosin. So when the calcium binds to the troponin, the troponin will turn the tryptomyosin around, exposing the myosin binding site. So normally the myosin binding site is hidden under the tryptomyosin. So the troponin will turn the tryptomyosin around, thus exposing the myosin binding site. So the myosin, this one is the myosin, can bind to the the myosin binding site and pulls the actin towards the center and and then eventually cause a contraction of the muscle. Okay, so this is what will potentially lead to the muscle. So the overall view, what it does is the GQ protein from the alpha-1 receptors, it contracts the smooth muscle in the blood vessels, thus causing the vasoconstriction, and then increasing the blood pressure. It also contract the muscular dilator pupil, and the pupil will be dilated. Okay, so there are three types of G proteins, right? So first, GQ protein is already done. Now we talk about the GS and the GI. GS means the stimulatory, I means inhibitory. So as the name shows, GS stimulates the AC. Okay, so GS stimulatory G protein is found in the beta-1 and beta-2 receptors. So when the beta-1 and beta-2 antibodies binds to the receptors, it's the same. It releases GTP, which initially binds to the GS. We release the GTP from the GS, and then the GTP will bind to the GS and activate the GS, and the activated GS will activate the AC and the new cyclase enzyme in the cell membrane. So the activated AC converts ATP to CAMP, which is a secondary messenger. And then it will also activate the protein kinase, which will lead to the phosphorylation of enzymes and initiate chemical responses. Okay, so this is the effect of the stimulatory G protein coupled receptors. So now let's move to the GI. As its name shows inhibitory. It inhibits the AC, the adenyl cyclase enzyme. So for example, this alpha-2 receptors. Okay, alpha receptors. When the neurotransmitter is releasing too much from the vesicle, it will bind to alpha-2 receptors, and then it will inhibit the AC enzyme, and then the ATP inhibit the enzyme, so the AC enzyme cannot work, so the ATP cannot be converted into the CAMP. There is no activation of this protein kinase, there is no fusion enzyme, so as a result there is no cell responses. Okay, so this is the overall view of the G protein coupled receptors in terms of pharmacodynamics.